Understanding cell metabolism is a concept that is still not entirely understood, but label-free multiphoton microscopy is a non-invasive way of monitoring cell metabolism and lipid production. This newer, non-invasive way of monitoring provides a direct quantitative numer it provides direct biomarkers that are noted, and they give off the adipogenic cell differentiation, and they tell you them the metabolic state. The first people to discover this was two Germans who studied at Cornell University in New York in 1990, Winfried Denk and James Strickler. They were the first people to do photon fluorescence and give it a 3D structure itself. They soon learned that the fluorescent emission increased with excitation intensity. So the fluorescence and the photobleaching were confined to the vicinity of the focal plane for the photon fluorescence. They had also used a confocal microscope, which is known for blocking out certain structures and being able to focus in more and create a sharper image. So then they created a one photon excitation fluorescent machine, two photon and excitation fluorescent machine, and then finally a multi-photon fluorescent machine. So a multi-photon is the combination of a two photon excitation fluorescence and a confocal microscope to create the multi-photon microscope. And combining these spatially resolved photochemistry is spatially resolved the photochemistry and the photolytic, and it showed us the photolytic release of caged effector molecules that were not being noticed before. On the far left is one photon excitation fluorescence, and it was only used for a little bit. They then moved on to the two photon excitation fluorescence, which is the orange one to the right. And as you can see, it's a lot smaller of a pinpoint versus on the left with the one photon, it has a lot more of a divergent beam. So there's a lot more of the X-rays absorbing into different areas. They did use an alternative to the confocal, but it was limited and it didn't give great detail at all. So on the far right is the multi-photon laser scanning microscope itself. They are now able to pinpoint a lot better and reject out of focus fluorescence and unblur the image a lot. So the difference is that the two photon excitation fluorescence machine was better about studying and evaluating epithelial tissue without needing a stain. Whereas the multi-photon microscope uses this optical redox ratio that computes numbers based off of the intracellular concentration. So still the dynamics are not 100% understood for cellular metabolism, but it's the step in understanding how the longevity of each of them works. So to the left is an example of a monkey's kidney tissue sample of underneath a confocal microscope versus the multi-photon microscope that they were using. As you can see with the left, there is very little detail. There's not good spatial resolution, and the confocal had a lot bigger of, it had more divergent beams. Versus the one on the right had the multi-photon, and you can see a lot, you can see the dark spaces, and you can see more detail and better contrast and spatial resolution. The ability to then pinpoint and reject the outer spaces then move them on to fluoroimaging, where they were able to add in the real-time spectrum, where they could pinpoint proteins and give them a certain dye and color, and then be able to watch these reactions, such as this happening, which is in a cerebral cortex. Wondering. And now it is so detailed, it almost looks like it's fake. They have a great spatial variability, and you can see the differences between the cell arrangements, and so you can tell what their cell functions are. And they now use this modality to look at the cytoarchitecture of specific areas, due to the fact that they're able to watch it in real time, and then they're able to pinpoint it with a protein, and then use fluoroimaging to watch the pathway. Opened a whole new world to diagnostic imaging. Developmentally, it is still only tested on animals, fortunately. But the best part is that there is such a better understanding of cell arrangement and their function. And the use of the ultra short pulsed laser with a near infrared emission, it gave off a lot of radiation. So just doing it on mice, while it's still something that is very new and not well understood, is the best that can be done currently. And they usually require multiple scans, so that's just a lot of radiation that keeps going, and we all are told about ALARA that any contraindication is if they're going to get too much radiation. These exams are usually performed below 10 megawatts. So the common feature aids the use of a high numerical aperture objective to get a high concentration of laser power inside the sample itself. The multimodal microscope used the near infrared fiber lasers, and these gave out 4 spectrally separate signals. And then the confocal microscope pinpointed from those signals where they were able to find specific dimensions, and that's what helped create that 3D portion of it, versus just a conventional microscope, just an everyday use microscope, everyday use, like we all use them. There is a secondary fluorescence that's emitted, and so it blurs out, and they can't get 100% the exact cell and part itself isolated. For patients, they are able to look at this data in real time. Through the multiple scans and then being able to pinpoint certain proteins and then create these 3D structures, doctors are better able to diagnose and track their patient's progress, so that they're able to change their care plan as needed, help their medical care as needed, make any changes as they need. These time-lapse experiments and real-time image acquisition for the cellular biology is very important information for the doctor, so always appreciated. The contrast and definition is very greatly improved due to the addition of the confocal, and living specimens can be examined with such good clarity. We are able to do different experiments and such on different animals, and we have a different level of clarity throughout the cells and understanding how they're reacting to certain things. And the overall just understanding of metabolic processes of humans. The human body is still evolving and changing over time. We will begin to lose our wisdom teeth, get extra bone in our foot, arms are longer. And as the technology advances with it, there needs to become the ability to also diagnose specifically what's going on with these other changes. And as this information is able to become more widespread and more able to be used by other people, things such as the label-free multi-photon microscope can aid in better diagnosing patients and creating a better environment for doctors.